tarde, espero que todos estejam bem. Eu sou Rosemeire Barbosa, coordenadora sociocultural do Grupo de Estudo Ganho Genético. Sejam todos bem-vindos, já agradeço a presença de vocês que nos acompanham aqui pelo canal do YouTube e aqueles que ainda não se inscreveram no canal, façam sua inscrição e ativem o sininho para ficarem por dentro de todos os nossos eventos. E hoje, dando sequência ao ciclo de palestras sobre os avanços tecnológicos no melhoramento de plantas, teremos a ilustre participação do Dr. Jose Crossa. E para auxiliar neste importante evento, contamos com a participação do Dr. Gabriel Gonçalves como moderador. O Dr. Gabriel Gonçalves possui graduação em agronomia e mestrado em recursos genéticos vegetais pela Universidade Federal de Santa Catarina, doutorado em genética e melhoramento de plantas pela Universidade Estadual do Norte Fluminense Darcy Ribeiro, realizou estágio sanduíche com bolsa PDSE da CAPES, sob supervisão do Distinguished Scientist do Centro Internacional de Melhoramento de Milho e Trigo CIMMYT, Dr. Jose Crossa, na unidade de Biometria e Estatística, e atualmente é pós-doutorando nota 10 da FAPERJ, com o trabalho de obter Dr. Gabriel.
Obrigado, Rosemeire. Boa tarde a todos e todas. Primeiramente, gostaria de parabenizar o Núcleo de Estudos em Ganho Genético, pela construção desse ciclo de palestra, com convidados de altíssimo nível, e agradeço o convite para participar aqui como mediador desse webinar, com a participação do professor Dr. Jose Crossa. Dr. Crossa é agrônomo pela Universidade da República Oriental do Uruguai e Ph.D. em Estatística e Genética Quantitativa pela Universidade de Nebraska-Lincoln em 1984. Possui um extenso currículo de produções científicas e premiações. É pesquisador do Centro Internacional de Melhoramento de Milho e Trigo, CIMMYT, na unidade de Biometria e Estatística. Também é professor cátedra do Colégio Pós-Graduados no México. Em 2008, recebeu o título de Distinguished Scientist como melhor cientista do CIGIAR, o Consultative Group of International Agricultural Research, ou Conselho em Pesquisa Agrícola Internacional, o qual o CIMMYT está inserido. Foi premiado pela Divisão de Conservação de Recursos Genéticos da Crop Science Society of America, com um dos 3 melhores artigos publicados na Crop Science nos anos de 1997, 99, 2002, 2004, 2005 e 2006. Em 2019, Jose Crossa foi reconhecido pela Web of Science Group como um dos pesquisadores mais influentes em sua área de atuação. De acordo com o Google Escolar, Dr. Crossa possui aproximadamente 33.700 citações, um índice H de 90, ou seja, 90 artigos com mais de 90 citações, e um índice I10 de 358, ou seja, 358 artigos com mais de 10 citações. Ele é membro da Sociedade da Agronomy Society of America, Crop Science Society of America, Mexican Academy of Science, Statistical Society of America, the International Biometric Society e Consultative Group for Agriculture Research of University of Sao Paulo. E é professor adjunto da Universidade de Nebraska, nos Estados Unidos. Foi do corpo editorial de diversas revistas científicas, como Crop Science, Eufídica, Agrociência, Revista Mexicana de Fitopatologia, e atualmente é do corpo editorial da revista Theoretical and Applied Genetics, The Open Horticulture Journal e The Crop Journal. Desenvolve pesquisas com análises genômicas e modelos para interação genótipo-ambiente, fenotipagem de alto rendimento, predição genômica para rendimento de grãos e resistência a doença em milho e trigo, além de ministrar curso de genética e estatística. Em 2018, publicou, junto com Jesus Seron Rojas, o livro Linear Selection Indices in Modern Plant Breeding, que é uma grande contribuição dos autores e do próprio CIMMYT para o melhoramento de plantas no mundo, e trata-se de uma publicação Open Access na renomada editora Springer. Hoje, o Dr. Jose Crossa irá apresentar a palestra intitulada Theory and Practice of Genomic Selection with Phenomics and Environmics. Good afternoon, Dr. Crossa. Thank you very much for accepting the invitation. The audience is yours. You can share your screen. You have to open here.
It is all right? Can you see my screen?
No, not yet.
Now?
Not yet.
That's strange, but I'm sorry. There is a problem here.
Okay. Just a little bit.
Okay, let's see. I have a connection problem right here at home. So let's see. I will start from zero, right? I will share. Sure.
Now we can see.
Okay, thank you. All right, thank you. Thank you, gracias, obrigado for the invitation. I will talk in English. And I'd like to present this to you, students and audience. Models and methods in genomics and phenomics. Not exactly in wheat breeding in maize and wheat, because CIMMYT is maize and wheat. So basically, I will go through. I think that I'm going to have more slides than time, so I try to go fast and perhaps avoid, or not avoid, but go fast across some slides which are not very important. I thought that to give you an idea on what do we do when we define target population of environments in breeding, what do we do with the sparse testing, and everyone talks now in sparse testing. What do we do? How do we integrate image analysis, multi-trait and multi-environment, and a little bit on deep learning, artificial neural network. Some of the slides you might have seen, perhaps, some of the slides I show, because it is not possible to change all the slides for every talk I give, but I hope you will enjoy and you will see what do we do in CIMMYT and at the Graduate College in Mexico. So the motivation is basically that the field phenomics and genomics offer an opportunity to accelerate breeding. We link genotype and phenotypes, and we try to link genotype and phenotype, but it's still a huge challenge for an optimal application of high throughput genomics and phenomics. We usually talk about genomic prediction based on a single trait, but we need to talk about multiple traits because we measure net genetic merits of the individuals. So we will see models, method designs, multi-trait, multi-environment, and see allocation designs, the GBLAB and the GBLAB by environment interaction. We always incorporate environment by genomic by environment interaction. In plants, the G by E is a crucial phenomenon. We will see functional data analysis, Bayesian multi-trait, multi-environment, and deep learning. In breeding, you see that in breeding, we try to put our product or the breeder's product as soon as possible in the market. And we do this up to now based on two sources of information, phenotypic and pedigree. The question is, can we do better with more information? Can we do better breeding, not only with phenotype information, the observation that we made in the field, with the pedigree, which is the genealogy, but using other information, the weather information, the climate information, the image information, the molecular information. Can we do better? Can we accelerate the time in which we breed a variety and the variety reach the farmer? Can we do that with the new tools? And what is the motivation? The motivation is that we have this, let me change the pointer. We have this equation, which is a response to selection. The response to selection is directly proportional to the intensity of selection. This C, which is the proportion of the additive genetic variance that goes into the selection, could be full sieve, half sieves, could be 1/2, 1/4, etc. Number of years, which is the interval, and the square root of the phenotype. So this is important. The main factor here is the time, and we try to reduce the time. By reducing time, we increase the prediction accuracy. We increase the velocity of the cycle. If we use 5 years to get a hybrid in maize, we try to do it in 2 years or 3 years. We go through the foundation of the breeding. We will see the Fisher, how Fisher formulate the average effect of an allele. This is the crucial quantity that we measure. The average effect of an allele on what? On the mean of individuals that inherit that allele. And this is the breeding value, which is the summation of the average effect of all alleles pertain to that particular individual. And we can give this foundation of breeding by the Fisher infinitesimal model. If we consider U as the breeding value, which is a linear combination of random variables, random loci, k loci. A is an allele that adds the fixed quantity A sub k to the breeding value. So we have the breeding value, we have the breeding value for each of the markers, for each of the genes that make up that particular individual, right? And the M genes, they might take the value 0, 1, 2 if they are homozygous or heterozygous. We assume that they are mutually independent. They are not. We have linkage equilibrium among genotypes, but we have linkage equilibrium which will slow down the rate of convergence. So when we increase the number of markers, then U, which is the breeding value, converges to a Gaussian normal distribution. For when we have a finite, when we have a nu which is large, then we have a mean and you have a variance. And when this k, when we have a large number of genes, then the breeding value can be assumed that has a multivariate normal distribution with mean mu and variance sigma square u. And this is the prediction of the breeding value based on molecular markers. That we say this is a genomic prediction. And this is basically what Mewisen did when recently, well, not recently, about 10 years ago, when the markers became cheaper. They become cheaper and therefore they become very dense. So we can sample the genome. So we have the effect of the alleles, we have the marker effect, and we add them up. This is like mu. So this is mu. This is mu, the previous mu on the previous equation, but now we call G. This is the genetic value of the individual, which is the summation, the additive effect of the SNPs, the markers, which are linear, we add them up, and is the foundation of the breeding, the Fisher infinitesimal model. So the prediction of the breeding value based on molecular markers is called genomic prediction, okay? So what is genomics, phenomics, and environment? We have here known genotypes, phenotypes, unknown genotypes, unknown environments. We have the DNA of these individuals under normal conditions, under stress conditions, drought, under heat, under different conditions. So, but we see the plants, here we see the plants, and we have these plants or animals genotype. The progeny of these plants, the seed that we harvest, is here. But the seed, we don't plant the seed. We genotype the seed. We predict the progeny of these individuals. And these are the seed of the progeny. But we don't go to the next cycle, which is planting and then evaluating this. We will predict this because we have markers here and we have markers here. So we have seen the parents, but we don't see the progeny. And we use an equation here. We use an equation which will be the effect of the environment, the effect of the genomics, and the interaction. All this is the genomic, but we can have the genealogy of these plants. We might know the relationship between these plants, so we might have the pedigree. So we have this information and we have this information. So we can make a prediction. We coverage the whole genome with markers. We assume that there are some linkages, equilibrium between at least one marker and one QTL. We don't need a threshold for the QTL. We use all QTLs. But we confront a lot of complexity, as Dan Gianola and Gustavo de los Campos and many others have clearly delineated. Why? Well, because we have a lot of environments here. We have a lot of markers here. We don't know where the genes are. Well, I mean, the genes for a complex trait are everywhere in the genome. So it is a complexity, and we have a large number of markers, and we have a G by E affecting all the results. So the prediction is not a very simple one, okay? So, but this is the basically the core of the genomic. We save time by not planting the seed and predicting them. That means that we don't need to phenotype anymore. No. I'm talking about 1 generation, 2 generations, 3 perhaps at the most. So be clear. I'm not saying that we don't need to phenotype anymore. No, never. We phenotype. The only thing that we are doing is just to try to save this particular generation of being in the field. So we save perhaps 3, 4, 5 months, half of a year, or 1 year. So this, I will show some equations because we need to incorporate, we always incorporate into the genomics, we incorporate the G by E. So we have, this is the basic model. We have the response variable, the environment, the lines, and the line by environment. The only thing that we have done. And the first publication incorporated in GBAE was by Juan Burgueño, and then Diego Jarquín, 2014, Eslot, 2012. He uses a different approach. We say, okay, let's replace the environment by the environmental covariables. Let's replace the lines by the genomics and the environment by line by genomics by environment interaction. Then we make assumptions and we say, okay, the environments are related by this matrix omega. The genomics, the individuals are related by the G matrix, which is the genomic matrix. And then we have an interaction here between the genomics and the environmental covariable. That is in case we have environmental covariable. We might not have, so we have a simpler model. So the G is the XX prime, this is the markers, this is the genomic similarity, this is the environmental similarity. So we have the models based on genomics, or we might have the models based on pedigree. So this is the strategy that basically Diego Jarquín uses for defining genotype by environment interaction. But our goal is borrowing information between lines as much as possible. If the lines, they have a genetic similarity, or if the environments have an environmental similarity. We will see this later on the envirotyping. So one of the first important issues that we need to solve in breeding is defining target population of environment. How do we define this? How do we define our population of sites that we breed for? And we have in the WIT program in CIMMYT, this is clearly the South Asian environment for CIMMYT breed is very important. So I will show you how do we define this TPE. Because the TPE is a variable group of current and future production environment. There are climatic variation in the same farmer's field that will change year to year, but we try to define target population of environment. Someone say, I breed for drought, for environments in drought. I breed for optimal environment. I breed for nitrogen, low nitrogen environment. But we need to define how many sites and where are the sites on that. So we did this in WIT, and we defined the factors affecting the target population of environment. The climate and the soil, hydrological characteristics, socioeconomic characteristics, seed production, all these are defining factors, affecting factors of TPE. Now we need to use a methodology for grouping locations and farms into TPE. We might use a simple stratified hierarchical cluster analysis, or might use a more sophisticated machine learning, random forest, or others to select samples of environment. Now, the selected environments are the environments where the breeder does the selection of the line. But then we have the target population of environment, which are different. Those are the environments that the breeders is selecting for. So we need to see how we can classify the environment in one target population of environment. But how do we define the target population of environment? Well, CIMMYT has basically breeding for 1, 2, 3, 4, 5, 6 type of different environments. Irrigation, optimal environment. Under bed. Bed is a planting system. Flat, optimal environment. 5 irrigation. Drought, drip. This is drought. 2 irrigations is about drought, but a little bit not exactly drought because you have 2 irrigations. But we have late heat and early heat. These two defines are very important environments in South Asia. I'm talking about South Asia because this will include many different heat stress phenotyping, and we have Obregon site which will simulate these actual environments that we will find or the breeder will find in South Asia. So we need to simulate these different environments. So there is a history of CIMMYT defining target breeding of environment. On the 70s, they classified environments into agroecological zones. Then these environments were reclassified by using more data collected during all these years in different zones of South Asia. And then CIMMYT defined a mega environment classification. That was around the 90s. Always trying to classify the mega environment. And then we start using the GIS tools to redefine the mega environment. That was around 2007. And now at this moment, we try to use big climate database to perform a cluster analysis, and this is historical databases and other methodologies for defining mega environment and target population of environment. So you see that since 1970 up to now, we follow the new technologies that we have for collecting data and for collecting climate data. So we did this. We started doing this in 2020 and finished 2021 after a year and a half, in which we define environment. So we define initially environment for India, by using this elite spring wheat international data. And we collect and we have environmental data for 16 years. We collected data for 245 site-year combination. We have meteorological data, minimum temperature, maximum temperature, relative humidity, soil data, very important soil data, soil water capacity. And then having this data, we classify the environment. We did a pattern analysis, we did a cluster analysis, and then we did we calculate direct, indirect response to selection, and we did a prediction based on pedigree of these three models, and see how the data in Obregon predicts South Asia. So we define basically these three target population of environment. High yield potential, that we call TPE1, heat stress, heat and drought stress. Okay? So this is TPE1, TPE2, this yellow, and TPE3 is and TPE3 is this vast central South India. And these are the sites that where we classify this site based on the environmental covariables, based on 16 years data of these sites under the testing that we have. But then we did the prediction based on pedigree. And you see that doing the prediction, we were able to predict the sites under 5 irrigation, 2 irrigation, late heat, 5 irrigation optimal, and drought. And we see that when we use the full model, which means the environments, the additive, and the additive by environment interaction, which is the green one, we have predictions which are about 0.5 for some sites, For drought, the prediction is much lower because it's much more difficult to predict the sites under drought or even under late heat. So we did the prediction using the data from Oregon. We predict the sites belonging to these environments here. So we ended up with a mixture of different of the site of the selection environment sites in Oregon, predicting different TPEs depending on the prediction accuracy. And this is what we expect to have as a genetic gain for grain yield from 2001 up to 2016, based on the data that we have, based on this SWIT, Elite Spring Wheat International Trials. So we see that for the different TPs, we have these different genetic gains or genetic trends. For some of them, the gains are very good. For others, are not that clear. But for this, and for this, and for this, the genetic gains are clearly. These are the populations that we tested, and these are the years tested. So you see that there is an increase on genetic gains. So that means that our classification of the TPE were done more or less according to what I hope we expected to have that. So that we published this paper in Frontier about a couple of months ago, because we believe that by doing this, we update our initial definition of mega environments done by CIMMYT with several years ago, and update and using all the resources we have and all the data we have to redefine this and to update this and to have a more clear picture on how the environments are clustered. Of course, that now with the climate change, year-to-year variability is much more difficult to assess. Okay? So this is for the target population of environment. Now, we will go on I'll give you an introduction of high throughput phenotyping. In maize and wheat in CIMMYT, in maize in Africa, but in wheat in Mexico, we have high throughput phenotyping, data collected with drones and airplanes. Okay? So we have data from different stages of the crop. So this is the value. So we know, at least we have an image of the early stages of the crop, medium stages, flowering time, post-flowering time, and we can use this image. How do we use the image collected by high throughput phenotyping? Well, we have reflectance data. We suppose we measure, under certain reflectance technique, we measure, let's say, 250 wavelength bands at certain range of the spectrum of the light. So each of these curves is a width line, and these are the wavelength. So we measure for each of the lines, we measure the wavelength. Conceptually, each curve represents one data, but the data here is not one point. The data is a function. The data is the response data is the function at a given point and under certain environmental conditions. So we have the environmental conditions that prevail when we took the photograph, the image, and we also find the given point time of the growing crop. So there are variability that we need to control. So we use on Oswald Montesinos and Lalo Montesinos, they decided to say we will use functional regression analysis. Why? Because we have the predictors, which are functions, and we might have the response, which is a function as well. So the functional regression can provide yield predictions with similar and in some cases much higher predictive power than the conventional regression techniques. Can we integrate the functional regression analysis to the models, the genomic models? Can we integrate this? Can we make a Bayesian approach using these models and integrate those with the genomics? Well, let's see. It is possible, and we have done this and published a model where we use high throughput phenotyping and we genomics or pedigree. For example, for a conventional linear regression model, we have a summation of wavelength, right? So these are the predictors, the functional predictors, and these are the functional linear regressions. Is the beta sub k is the beta regression coefficient for the k band that will be estimated. So what we have done is just to use this summation from k equal to 1 up to 250 in the genomic model. We have here the genomic model environment G and G times E. We added the conventional linear regression to try to model those wavelengths. Okay. The problem is that these are not discrete; these are continuous. So if they are continuous, we need to use a continuous variable. If we have 250 wavelengths, are functional covariables that we construct them at an interval between certain limits of the wavelength, which are the minimum and the maximum, at which each of the 250 wavelengths bands reflectance are measured. So we transform the summation into an integral. And that complicates a little bit more the problem, but we can still handle that. We need to do some mathematical manipulation to see how do we do this integral and to see how to incorporate this into the Bayesian analysis. But this is what we have done using this functional regression analysis. And then these are the results of using a certain number of models at a certain time point, 2, 3, 4, 5. This is early stage of the plant. This is intermediate, and this is by flowering. And these are the models that give the best prediction is when we assess the wavelength by environment interaction and we put genomics. And we have this for 3 environments: drought, irrigated, and reduced irrigated. But you see that for the 4th time point, we get, for example, this is early stage of the plant. We still, when we use an appropriate model, we increase the prediction accuracy up to 0.4. Early. If we use wavelength in the appropriate statistical methodology, which is assessing the wavelength by environment interaction, of course, because it's not the same to the climate in one place is not the same as the climate in another environment. And because there are the humidity of the air, the temperature are always affecting the wavelength. So this model from 9 to 14, they have this and they are always give an increase in prediction accuracy. So genomics and high throughput phenotyping, genomics and phenomics work all right. Other results from genomics is that Margaret Krauss, when she was in CIMMYT, she published this in 2009. And I'm sorry for this. This is a very heavy picture, very busy picture. We have the years. We have 1, 2, 3, 4 years. We have the colors here are the different growing stages, vegetative, heading, grain filling, and all. And what Margaret did here, he said that he developed a metrics that use the relationship between the wavelength, right? The bands. So the model we used was a genomic model, and then we put the wavelength by environment interaction. So we have genomics, wavelength by environment interaction. And we have each of these growth stages for models with only G, with only A. Remember, A is a pedigree, with G plus A, G plus G plus E interaction, A plus E, and then the model with the wavelength. And then the models, including the interaction, the wavelength by the genomic plus the wavelength by environment interaction. And you see that when we increase interaction, there is always a tendency to increase the prediction accuracy. These two, these two, these two, and these two. There's always a tendency to increase the prediction accuracy when we include the interaction effect. So again, this is another result that says that we can join together genomics and phenomics, use an appropriate model for genomics, use an appropriate model for phenomics, and all together, like using the wavelength, have this increase in the prediction accuracy. And we have this for moderate drought, severe drought, and heat. Okay? So this is the results, phenomic genomics. Recently, Roberto Fritz Neto, with his student Germano Costa Neto, did a very good study on genomics and environmics. Because how to use environmental covariables is another important issue. Remember that we have the phenotype. The phenotype is the genotype, the environment, and the interaction. The phenotype is the genotype, the environmental covariables, and the interaction between the genomics and the environmental covariables. Remember that genomics, we say genomics, but in genomics we have the additive and we have the dominance. So we have an additive by environment and a dominance by environment. But we also, and Germano also developed a technique to use environmental covariables, collected large number of environmental covariables, but also use functions of the environmental covariables as we use function of the markers. Because we collected a lot of environmental covariables. We collected the climate data and we collected the soil data, and the soil data is very important. So Germano was able to put results on that and use different type of relationship metrics. So the model that he uses is environment, additive, and dominance. Environment, additive by dominance, plus genomic by environment. Okay? The genomic by environment is the additive by environment and the dominance by environment. Then we added the environmental covariables. Instead of putting environment, we added the environmental covariables. So we need to model all the environmental covariables and genomic by environmental covariables. Now the G by E includes the G, well, I guess this should be A by, it should be A by environment and D by environment. There is a mistake, I guess, here. And also G by W, which is additive by environment and dominance by environment. And we use the GBLAB. We use a Gaussian kernel, which is a nonlinear kernel that helps us to improve the prediction accuracy, and also a deep kernel, which is also a nonlinear kernel. And you see that the best predicted model was always the one that include the genomic by environment interaction, additive by environmental covariables, dominance by environmental covariables, which is blue, and the dash, which is the Gaussian kernel, which is most of the time giving the best prediction across most of the environments. That again is a proof that genomics and environment is a very good business to do. The problem is that environments are complicated if we have so many years, so many sites, and we need to model that, and we need to be smart to take a good environmental data to be able to improve prediction accuracy. So the question that we ask, and many people ask, is there a way to have a climate smart solution for environmics and genomics? Well, I think that we can, but we need to start thinking in representative phenotype information, which are the envirotypes that limit the adaptation of the crop to some breeding TPEs. We need to understand the relationship with the secondary traits. You know, it is in reality a solution that we can present, but takes time to arrange the data, to use the data, and especially with the climate change, and everything changes year to year. So the prediction of next year is very difficult to make. Another approach using genomics and environmics was the one recently published by Gustavo de los Campos. Gustavo said, we have in the usual network of trials of breeding, we tested the lines or the genotypes, the lines with the genomics. We tested only a few years, only 3 years at the most. But we have data from environmental covariables, we have data from phenotypes, and we have data for genotype, for genomics. So we use a Bayesian model to predict within this particular site. This is the north of France, or France, most of the points are the north of France. But we have each of these points, we have a lot of environmental covariables. What do we do with the virtual network of trials where we have historical data collected in many of these sites? We might have 10 or 20 years historical weather data that we can use. So the question is, can we sort of do a data-driven computer simulation study where we are going to use this data and see how is the realization of these genotypes under these historical weather conditions? Can we make can we see the uncertainty existing when we predict these genotypes under these historical weather conditions. So we have historical weather records at target locations. Yes, we can, and Gustavo shows that, and he shows that there is we integrate into the model all this uncertainty. And these are the uncertainties of each of the lines here in only four environments than that. So we can predict the performance of the genotypes in these conditions, but remembering that we have uncertainty, that we need to compute this uncertainty. We have a distribution here. It's not one value. The prediction is not one value. We usually take the mean of this, or the mode of this, but there is an uncertainty that we need to consider. And this is what they have done, but still the predictions were in reality very good in those environments that they were never tested. Okay? Now, let's go to why multi-trait and multi-environment. Why the deep learning multi-trait, multi-environment? Is this because it's fashionable? No, it's because we need to go to multi-trait, multi-environment because we like to see the net genetic merit of an individual when a lot of traits are measured, and many of the traits are related to each other. So most of the time, we are better off doing a multi-trait analysis, a multivariate analysis with multi-trait than single-trait analysis. And of course, when we do this multi-trait, we can also do the multi-environment. Why the deep learning? Because it's fashionable? No, because I think that when we have multi-trait, the deep learning has some good, very useful function that can put together different traits at different scale, and this may have an advantage over the standard conventional analysis. I'll show you some of these results. This is with Oswald Montesinos and others. 2016, we published a model which is Bayesian multi-trait, multi-environment. We have the fixed effect beta that includes a trait by environment interaction. We have a random effect B1 that has a trait by genotype. And we have the random effect B2 that includes a trait by genotype by environment. This is a very, very efficient model because its Bayesian is slow. You need to be patient when you run this. We have a publication in 2019 where you can see some of the R code that we use for fitting this multi-trait, multi-environment model. We know that Bayesian multi-trait, multi-environment model, for many different traits, we get a higher correlation using the unstructured Bayesian multi-trait, multi-environment than using the univariate. For example, well, not for days to herin, but for NDPI 079, 033. For yield 064, 058, 064, 060. So you see that there are many traits that are better predicted when we use the multi-trait. That is one of the reasons why this is one of the reasons. The main reason is that we like to see the net genetic merit of the individual and have the multi-trait and the GBB for each of the traits under these conditions. We use this genomic-based prediction of multi-width quality traits in multiple years. For example, data 1 has so many lines tested in this year. Out of these, 658 lines were tested in 2014, 2015. Can we predict this using this? Can we predict this 686 in this year using the previous year? Can we predict this using the previous year? Can we predict this using the previous year? And so forth. We use the Bayesian multi-trait multi-environment and the multi-trait rich regression. And we predict this. 5 years data, a lot of data. These are the results. We have Bayesian multi-trait multi-environment green and multi-trait rich blue. We see that for the correlation, we see that most of the time, for most of the traits, the green one, the Bayesian multi-trait multi-environment, is better than the rich regression. This is another measurement, which is the opposite of this. This is the larger the better. This is the smaller the better. The smaller is the green one. For this 3 years, and we have the same for these 2 years here. And the MAPE is the mean arctangent percentage error between what is forecast and the eventual outcome. So the smaller, the better. But again, it's a clear advantage of the Bayesian multi-trait multi-environment. And many of the traits are very well predicted. Very, very well predicted, 0.6. These are quality traits. Be careful, these are quality traits, which are easier to predict at grain yield. Now, why artificial neural network? Well, let's see if I can show why. You know, neural network, we inspire on the biology, on how the neurons are connected to each other. For example, all these, they join a function, but we can have different layers here. This is the input, these are the markers, this might be the phenotype data, then we transform the markers and the phenotype data, we make one transformation or two transformations, and this is the output, which is the predicted value. But it's based on the biology of the brain's response to external stimulus, okay? And we can have this for single trait. For a single trait, there is only one output here. This is for a single trait. This is for a multiple trait. And how this works? Well, for a multiple trait, this is what we have. For a multiple trait, this is the output of the input layer, which is the input of this hidden layer. Okay? The output, so the output of the hidden layer is the input of this another layer. But you see that we do transformations here from here to here. We do another transformation from here to here. We do another transformation of the data from here to here. And we just can use different functions in here to try to capture something between markers that we cannot capture on the standard GBLAB. This is the main idea that we have. Okay? Looks complicated, but in reality it's not that much complicated. It requires a lot of computing time because we need to tune the data of these models. And that is one of the limitations of that. We apply this in a multi-environment genomic prediction for one single continuous trait using deep learning and using the GBLAB. And we publish this using G/E or non-G/E. The deep learning is the red one, the GBLAB is the yellow, is the green one. You see that if we include into the model the G/E, the GBLAB is better than the deep learning. If we don't include the G/E, the deep learning gets very good, at least for some data set. This is maize and this is wheat. Why? Because the deep learning seems to be this is a machine learning, so it's a machine learning that learns what the data is telling us. So the deep learning is very sensitive to the different environments. So we don't need to put the G by E on the deep learning. We just don't say that there is a G by E. The deep learning is supposed to learn that. Okay? And actually, it learns in all these 8 data sets. 9 data sets. We included this wheat from Iran. The same with a multi-trait genomic prediction of continuous multi-trait. We don't use one trait, we use several multi-traits. Again, including G by E, not including G by E, the multi-trait deep learning is better than the Bayesian multi-trait multi-environment. If we don't put G by E, if we put G by E, the Bayesian multi-trait multi-environment overpass the deep learning. Okay. All right. So now, what I will show now? I will show now some of the results of using rapid cycling genomic selection in CMID maize for grain yield. There is our colleagues in Africa, Jose Begene, Mange, and others. They have 8 biparental populations. So they selected, they made rapid cycling, 3 cycles in 1 year, 8 biparental population. This is across 8 biparental population. This is cycle 0, cycle 1, cycle 2, cycle 3. On average, an increase of 86 kilograms per hectare per cycle. Biparental. Another colleague in Mexico, Shuekai Sang, he did this for multiparental populations. 2 cycles in 1 year. Very good prediction, very good increase in prediction accuracy. 225 kilograms per hectare per cycle. Very good results, published 2015, 2017. Results of Joseph Begene on rapid cycling genomic selection, grain yield. Like I said, this is one biparental, other biparental, cycle 0, cycle 1, cycle 2, cycle 3. There's a clear increase in cycle 3 over cycle 0. At least here, not much here, here, cycle 2, cycle 3. So we see that there is a gain. And the overall gain in grain yield is 70.5 kilograms per year per hectare that overcome other conventional genetic gain for conventional breeding in Africa about 2 times higher. That's the reason why at the moment in Mexico and in Africa, most of the breeding program is rapid cycling as rapid as possible, and trying to avoid stage 1 and go directly to stage 2, and using this in order to increase genetic gains. You see the performance of hybrid developed from lines developed through rapid cycling genomic selection. Most of the time, the top 5 hybrids are coming from lines developed under genomic selection for optimal and for drought. So a large number of lines developed under rapid cycling genomic selection as parents for participating in hybrids that are used in several parts of sub-Saharan Africa. So very good results, very rapid cycling. The same if you are not convinced that you can trust on markers, Again, Joseph Begene and Mange Goldman, they did this experiment. They said, well, let's make sure that the guys that we selected based on markers will give the same grain yield than those selected under phenotypic. So they compare hybrid selected and genomics under phenotypic. There are no differences on that. This is a commercial check. This is genomic selection. Those selected for the same crosses, of course. Those selected under genomics, phenomics. This is for managed drought. This is optimal condition. And this is the mean of the top. Mean of the top for genomics, mean of the top for phenomics, under managed, under optimal conditions. So the breeders are very clearly believing that markers can give the same prediction accuracy as they get using phenotype, at least for a couple of generations. And the same was done by Xuecai in Mexico, where he selected maize, white and yellow, white phenomic selection, white genomic selection, yellow, this is white maize, yellow maize, and yellow and white the top. You see that there are no differences between selecting based on genomics or based on phenotype. And finally, I like to say that we developed, well, not we, I would say Gustavo and Paulino, the BGLR package, which is the main machinery that we use for genomic prediction. Has a lot of power for using many different genomic selection genomic prediction methodologies. It does the G by E clearly. And recently, we developed with Diana, which was our student here at the Colegio de Postgraduado, Diana developed the LMF4 genomic selection, which is an R package for genomic selection. Because sometimes the BGLR is very, because its Bayesian is slow, Monte Carlo always is slow, the process and the computing has intense use, but the LM4GS is a little faster than this and does many of the things and most of the things that does the BGLR. So if you want to download this and try it, you are welcome to do so. So I like to certainly, this is not done by one person. This is done by so many researchers and students from CIMMYT, all scientists, my colleagues, ICRISAT. We have project with Norway University, with Graminur, which is the seed company associated with the university. We have project with the Swedish University of Agricultural Science. Of course, I teach and I am member of the Colegio de Postgraduado Mexico. We teach with Paulino genomic selection course. Montesinos Oswald is from the University of Colima in Mexico. His brother Lalo is from the University of Guadalajara. Of course, Gabriel was with us many months. We have a very close collaboration with Roberto and students that for years and years were coming to CIMMYT to do some research on genomic selection. We receive students from Embrapa, maize and rice in Brazil. We have Diego at the University of Lincoln, Nebraska-Lincoln. The University of Quintana Roo, West Jaime is there, a very good collaborator and researcher in Gaussian kernel. In Michigan State, which is Gustavo, that is always a source of inspiration for us because he does always very good research on genomics in plants. The University of Wisconsin, where Daniel Gianola, which is my first teacher in genomics, is there. In Wageningen University with Fred Wageningen, University of Hohenheim, Germany with Hans-Peter Fifo. We always collaborate and talk. And UC Davis with Daniel Runzi is there. And the Cornell University, which is we work with them very, very close in the initial projects. So all these are the collaborations that we have in order to come up with some new things that we believe are important. And finally, this accelerating genetic gains based on WIT, which is a new project that basically give us the opportunity to try to do this rapid cycle in genomic selection and shortening the cycle and see what will happen. We hope that we are going to get very good results and show that genomics is a good system to apply in breeding for accelerating genetic gains and the cycle. And thank you for your interest. And if you have any question, comments, I will be glad to answer this. And thank you very much.
Gabriel, desculpa, você poderia ligar o seu microfone? Obrigada.
I'm sorry. Thank you very much, Dr. Crossa, for this brilliant presentation, for sharing a little bit of our knowledge and the CIMIT work as well. Now, let's move on to the questions. We have a question from Jan Torres. Congratulations, Dr. Cross, for excellent lecture. I would like to know what are the challenges to work with multi-environment in view of climate changes. Thank you very much, Jan Torres.
Yeah, well, there are many challenges, right? Because whatever you have this year may not happen next year. So that's a big problem, because that's the reason why we believe and basically what Resendes, Roberto, Germano have been doing in Brazil in trying to do some work on envirotype, because those are the ones that will help us to assess these challenges, because the challenges are big and complicated, you see, because it's very difficult to predict the weather in next year. We can use historical data, and we need to use historical data. And we need to use something similar to what Gustavo de los Campos has done in this publication in Nature Genetics in 2020 and 2020. Use historical data to see how will be the realization of the genotype that you have for these 3 or 4 years in another year, in another condition, and see how is the uncertainty of that. Because we need to live with that uncertainty. Right? We need to learn how to live with the uncertainty of not knowing what will happen, 2 or 3 years from now. But we have the past data. The past data can tell us something about the new data. Well, but the new data is the new data, so the new data is the new story, but we need to be prepared for that, and we need to at least to try to see how is the frequency of all these rare climatic events that are happening, for us to account for those and to improve the accuracy of the prediction based on these rare events. And the uncertainty that they have, of course. I think it's difficult question to answer, but.
We have another question from Janio Stachio. Dr. Cross, you showed for us that the dataset will increase year by year with phenotypes, images, SNP, environment information, and so forth.
Yes.
For a given breeding program with focus in the given region, what would be your recommendation for training set?
For a training set, I would say
There
yeah,
is more. Should we use a sub-data set to fit the predictive model using only related data, or should we use all data available?
yeah, that's another good question that we've been talking around this for years, but sometimes we don't know. We have in wheat, we have about 7 years data. We have 68 varieties, line varieties, about 9 to 10,000 each year. We tended to believe that adding years, suppose we predict 2021, 9,000, using 60,000 from the previous years. We tended to think that by adding years more than 3 years back, we did not improve the prediction accuracy, but we were wrong. I think that the more data back we use, the better the prediction. Not much, not much. I would say going from 0.29, 0.231, 0.23, 0.231, 0.232, 0.23, up to 0.38, more or less. So I think that now that depends on the population. Semi-population at these stages are not the families. The families are in reality not related. Well, they are related, but they are very small families with 2 or 3 individuals. So we cannot believe that we have family relationships in those, because we have families with 2 or 3 individuals. But we believe that we can increase the population size by adding more years and try to pick up some alleles or haplotypes that were back 10 years ago that are still there and are still a sample of the population that we want to train. So I would say that if you can use all data, use all data.
Okay.
Unless you have a computing problem that might not be, you might not use. But I will use all data because it's not going to hurt you. It will be good in case it's needed.
We have another question from Jean Torres. We know the environmics is the new approach in plant breeding. In this sense, what's the best way to study the climate factor, and what is in your crop model to predict genetic gain?
Yeah, that's a good question. We don't use. One of the problems I have with the crop model is that the G by E is fixed. They cannot put G by E as a random. They cannot put the G by E as a random effect when you use the crop model, and so that is one of the problems. What De Los Campos did was just to take a data-driven simulation and try to translate whatever happens on the atmosphere, what happened at the plot level, and that may improve the prediction accuracy. But you have to have also a very efficient way to deal with environmental covariables. You have to have an efficient manner to use this and to be able to run and to organize the data, because you have the soil data and you have the climate data. So you need to separate both and see what happened with the soil data and see what happened with the climate data. The soil data assess the static G by E. The climate data assess the unpredictable G by E. So it's because that is a year effect, and the year effect is difficult to assess. Especially for the CIMMYT guys, for the CIMMYT breeding program, where every year the varieties are different. It's not like a standard breeding program where you have some varieties tested 2 or 3 years. We have different varieties and different years. So the year effect is confounded with the variety effect. The only click on the connectivity is the pedigree or the markers. So that adds another complexity that we have at least in CIMMYT. But this is because the breeding programs in CIMMYT requires not to have, many of the genotypes are new every year, because CIMMYT needs to deliver new things to the farmers and to the national programs.
We have another question from Rosimara. Congratulations, Professor, for the excellent lecture. There are few plant breeding groups studying this topic, environics. Do you believe this topic will be the new trend in different breeding programs in the coming years?
The topics of genomic, phenomics, and environmics, yes.
Yeah,
It should be. Is he talking about this? If he talking about environmics or image?
Yeah.
Yeah, I think that those are the ones that we will have to use. That data we'll have to use. I'm not saying that we are not going to use observation data, because we use phenomics is observation, phenotypes are observation, but phenomics are the observation that we have in the field for the plant at different stages, right? We need to use that. Now, to me, this is not going to replace any human intervention. Absolutely, I don't think so. Perhaps the role of the breeder becomes more, more inclined as to what data will give, what data from what sites will be more reliable in order to include it in the training population, and what data will be for sites and for genotype or for lines, what data is more reliable for me to believe that this has to be the one that I select for next cycle. But also we have a lot of software for helping the breeders to make a good selection of the parents based on genomics.
While the audience are typing the question, I will ask mine. And you showed many terms to include in the model, and I would like to know what are the biggest challenges to fit a good model with all covariates and genomic metrics and images. Even more so in experiments under augmented block design, widely used in CIMMYT with genotype by environment interaction, repeat controls, and special covariance. What's the biggest challenge to fit this model?
Well, I guess computing capacity is the models themselves, the statistical models themselves, their limitations. That's the reason why when we have the data, we put the model first. we as a statistician, biometrician, we say, well, this is the model, assuming that the response variable has a normal multivariate normal distribution, mean zero variance sigma square. We find this multi-environment, multi-trait, but the new, the new machine learning, statistical machine learning, they also give a lot of importance to the data. So they feed the data not with a predetermined model, but with just a simple linear model. And then they start learning from the data that this model has to be changed here, it has to be changed there. So we learn from the data. So now is the time for us to change a little bit, not to change the hard work of the statistical theory, but for us, if there is not overfitting, the model, the best predictive model is the one that we will use. And sometimes it's this model, sometimes it's that one. But deep learning, artificial neural network, or other sort of topology for the models, they seem to be gaining a lot of importance in prediction. That's the reason why we are there, especially under complicated data set. So we need to have an open mind. If we don't have an open mind, I mean, that doesn't mean that we use any data and doesn't matter if it because you need to be careful. If you have a prediction of 99, 0.99, that's not a good prediction. That is a wrong prediction. I don't think that unless there is something with heritability equal to 1. If there is something with heritability equal to 1, then you don't need to do genomics, and you don't care about the environmics. You just select what you see, and that's it, and go home and take a beer or watch a soccer game. Not a Uruguayan soccer game, because Uruguayan always lose. But we need to be careful with that, because sometimes when we always commit mistakes, but especially at the initial stages, we might say, oh, my predictions are very good, 0.9. There are cases that might be like that. If there is good correlation between sites, you might have high prediction because the sites are correlated, and because the traits are correlated, and you borrow information from the sites and from the traits and from the genotypes. But most of the time for grain yield, the predictions, if you get a prediction 0.5 for grain yield, you are good. For maize and wheat, this is what I say. For others, I don't know, autotetraploid crops like potato, they might go higher. Barley, which is not complicated, genetically not very complicated, might go high than 0.4, 0.5. But in general, these are the prediction. If I find a prediction accuracy of 0.3, I will not be unhappy. I will be happy.
We have more questions from YouTube. I can do another question. I would like to know if in the routine of the breeding program of CIMIC, you are using this kind of analysis, or due to the need to move quickly with the genotypes, do you use mainly the basic analysis to advance?
No, we use, for doing genomics, you need markers. So we always use markers if we have, or pedigree. We have a good system, a good pedigree system. If you have a good breeding program, you should have a good pedigree system. You should have a good genealogy. You should know who is the mother and the father and the sons and the daughter and the progeny of your elite lines. You should know that. You should have a very good genealogy. And if you don't have it, start doing that and start taking notes on who is the mother and the father and the son and the grandfather and the grandson and so forth for your data, because this is important.
Thank you. We have one more question for Poliana Capobiango. Congratulations, Dr. Cross, for your excellent talk. In your opinion, what are the perspectives of the application of environmics in a breeding pipeline operationally thinking?
Oh, I think these are the perspective are very, very promising. Are very, very promising. Now you have environmental sensors everywhere, right? You can take data. Of course, you are interested in the environment that happens on the field that you grew the crop, right? Now, if your crop is in Brasilia, you are collecting data in Sao Paulo, tough life. But the environmental data has to be more or less close to the one that the crop is. But I see that in the future, I don't think that we are going to be doing breeding without environmental data. Environmental data has been the forgotten aspect of the quantitative genetic equation, because we say, okay, this is the environment, E. Okay, but E, what is E? What is the environment? What environment are you talking about? Are you talking about an environment in where? Are you talking about an environment, a high land, low land, drought, heat? What type of data are you collecting? You can collect data basically every hour, right? Every minute. But that the question, the problem is how do you relate this data that you collected in the climate data? How this is related to what happened in the field, in the plot, right? Because the plot is not going to change much. The plants in the plot are not going to change much unless there is a really extreme case of weather conditions, like a hurricane or a big rain or a big strong wind that might make lodging. Small changes in temperature are not going to change the response of the plants in the field. If you change one day to another 1 degree or 2 degrees, those are unnoticed. The challenge is how to translate what is happening, not in the soil, because the plant in the soil will reflect something, that's for sure, because the roots. How the plant, how the climatic conditions will affect the plants in terms of maturity, in terms of heading dates or maturity date or other things. That is a challenge I see. So environmic is there, for sure. Image is there for sure, because image, you can measure some diseases very, very clear, especially in maize, very, very clear. And you have a very good correlation between what you see in the field and what you have in your image. Very, very clear. And Francelino, I don't know if you know Francelino Rodriguez, is a guy that that I saw. Yeah, he's from Londrina. No, he's from Sao Paulo, right?
Curitiba.
Curitiba. It's not Fernando. I'm talking about Francelino.
Francelino, I think, is also from Curitiba.
Really?
I don't know.
Okay. He was able to take image analysis from a disease for now. I don't remember which one was the fusarium or something in maize. And the correlation between the image and the visual were 0.7 for those diseases that goes along the leaf of the maize. Very good prediction. So the visual prediction was very good. So we were able to do GWAS analysis using the visual prediction, do genomic prediction using the visual values that were correlated with the visual. The image were highly correlated with the visual. Therefore, the image were good predictors of what happened in the field. That was a very good study. To me, one of the best.
Okay, thank you. I think that we can close the question.
Okay, yes.
Okay. And on behalf of the Research Group of Genetic and Plant Breeding Postgraduate Program of Universidade Estadual do Norte Fluminense, Darcy Ribeiro, I would like to thank you again for accepting the invitation, for the brilliant lecture, and for answering the audience question. And Rosemary, the audience is yours. Okay?
Obrigado, Dr. Gabriel, pela moderação da palestra e condução da discussão. Thank you, Professor Crossa, for your participation and excellent talking. E obrigada a todos pela participação e interação no chat. Tivemos o professor Messia, que esteve por aqui prestigiando a nossa live. Os egressos do programa, Ismael e Jânio Eustáquio, também passaram por aqui, obrigado. E a todos, não esqueçam de deixar o seu like e acionar o sininho de notificações para não perder os próximos eventos. Além do mais, vocês podem nos acompanhar pelas redes sociais do Ganho Genético, através do Instagram, Facebook e LinkedIn. E já gostaria de anunciar o nosso próximo evento, a webinar com o Dr. Magno Ramalho, intitulado Meio Século de Melhoramento Genético na UFLA, Dificuldades, Sucessos e Desafios Futuros, que ocorrerá no dia 12/8, às 17 horas, no canal do YouTube Ganho Genético. Lembrando que as atualizações dos próximos eventos estão disponíveis no nosso site. Obrigada a todos que nos acompanharam durante a live. Aguardamos todos vocês para os próximos eventos. Fiquem bem.